Assalamualaikum and hello everyone. Welcome to this channel by me Mo. You are watching a video series about electrocardiogram. In this video, we are going to talk about another fundamental knowledge of ECG, that is the conduction system of the heart. If you haven't watched the first video on cellular electrophysiology, you can do so by clicking on the link here or through the description down below. The references and credit for this video go to Textbook of Medical Physiology by Guyton and Hall and the only EKG book you'll need by Malcolm S. Thaler. Alright, let us begin. This video is going to be a short one, so what I am going to do today is to show a diagrammatic presentation of how our heart's conduction system looks like. Previously, we have looked at the interaction at molecular level in the first video. Conduction system we are highlighting today are between the cardiac tissues, more precisely about how the SA node convey its signal to the rest of the cardiac cells. Since ECG is built on fundamental principles in physics, we can't really understand ECG without knowing this. Knowing how the signal are conducted throughout the heart also means identifying the electrical vectors. Other rhythmic abnormalities of the heart are also built upon this knowledge. Let's begin with the source of the signal, that is our normal pacemaker, the SA node. Our SA node are neither made up of nerve cells nor your typical myocyte cells. It is a group of specialized kind of cardiac myocyte cell. We have seen how unique this pacemaker is previously. And other than having its own rhythmic automaticity, it is also non-contractile. This group of tissue is only about 3 mm in length. Its individual cells are smaller than that of the other cardiac myocyte cell. It is located at superior posterolateral portion of the right atrial wall or inferior lateral to the opening of superior vena cava. From here, it sends signal via three pathways. The first one, it goes to the surrounding adjacent cells by gap junction. This wave of signal spread throughout the right atrium at a speed of about 0.3 meter per second. Secondly, it goes to the left atrium via the interatrial bundle, also known as the Bachmann bundle. And thirdly, to the AV node. The signal transmitted to AV node via three bundles collectively known as the internodal bundle, since it connects these two nodal tissues. These individual bundles are the anterior, lateral, and posterior internodal bundle. Similarly, it is non-contractile kind of myocyte cell. It transmits the signal at a speed faster than gap junction, that is about 1 meter per second. AV node is located at inferior posterior wall of the right atrium, rightly behind the tricuspid valve. Now, even though the speed at which the signal is transmitted to AV node is faster than a typical myocyte gap junction, this group of tissue needs to penetrate through a fibrous tissue barrier separating the atria and the ventricles. This will slow down the conduction here so much that it allows the signal spread via other pathways to depolarize the whole atria wall first before it can pass the signal to the His bundle. It passes the signal via small fascicles that squeeze in between the fibrous tissue. From here, His bundle, or also known as the distal portion of AV bundle, gives out to two main branches. They are the right bundle to the right ventricle and left bundle to left ventricle. Since the left ventricle is thrice the size of the right ventricle, then it is only fitting that it also has three branches. They are the septal, anterior, and posterior fascicle to the ventricular walls. Cumulatively, the His bundle and its branches are also called the ventricular Purkinje system. Likewise, they are also a group of specialized cardiac cells which are non-contractile. Unlike SA node, their individual cells are actually larger than that of the ventricular myocyte cell. They also conduct electrical signal at a speed of 4 meters per second, the fastest, almost instantaneously delivering them to all cardiac ventricular cells by having specialized kind of gap junctions with very high permeability. At the very end, they penetrate about 1/3 of the muscular layer, that is the endocardium. Lastly, something that is also worthy of mention is that at septal fascicle, they conduct signal in the septal wall in the direction from left to right. Now remember that because you will make sense of the ECG later with this. Now that we have mentioned about the speed of transmission in each specified bundle, let's see how all of these work out in a timeline. Our starting point, SA node, is going to be our 0. From here, it reaches the transitional fiber, that is the junction between internodal bundles and the AV node, after 0.03 second. The signal then travels in the AV node for another 0.09 second, so by the time it reaches the AV node, it is 0.12 second. Then another 0.04 second to his bundle, making it 0.16 second from SA node firing. The signal reaches the endocardial surface at 0.19 second and epicardial by 0.21 seconds. Now, the best picture I could find that could help us depict this is from ScienceDirect.com, from an article by David Brown right here. It shows all of the pathways for you to compare except the septal fascicle. For the complete ventricular Purkinje system, this is a picture from Malcolm S. Thaler book showing all 3 fascicles. Now remember, not only the pathways but their direction of signal or electrical vectors as well, as it will be useful for you when we start analyzing our ECG strip later. I'll see you guys again in the next one. If you're interested in other ongoing series such as the human anatomy according to its body system, you can click on the link here as well. If this video proved useful, hit the like button and share them with friends and family. Subscribe for future videos. Thank you for watching. Bye bye.